Hello, and thank you for joining us. My name is Dr. Kristin Hennessy-McDonald, and I'm the Outreach and Community Engagement Lead with Mini PCR. I'm here with Mini PCR Bio to introduce our newly updated Food Safety Lab, Food Truck Trouble, which we launched in the fall of 2025. Before we dive into today's lab, I want to start with an introduction. If you're new to Mini PCR Bio, we make biotech equipment and curriculum to make it easier for teachers to equip their students with the latest biotechnology tools. Our durable, portable, and most importantly, affordable equipment is used for a number of different applications. These include scientific researchers in the field, like scientists in outdoor research labs. We also have community members who use our equipment and reagents to do things like identify foraged mushroom species. And of course, students and educators in classrooms. At Mini PCR, our overarching goal is to put biotechnology tools in everyone's hands. Our curriculum team is a group of scientists and educators who are passionate about making biotechnology education accessible for teachers. You see our curriculum team here, all sharing a picture of something that brought us joy. As I said, my name is Kristin Hennessy-McDonald, and you see me there in the upper right when I was a teacher at sea on the NOAA ship Oregon 2. We performed a long line survey in the Gulf. I spent 12 years in the classroom teaching life science, from 7th grade science to general biology to AP and dual enrollment biology, and quite a few things in between. I joined Mini PCR Bio because I loved their innovative lab activities. As a classroom teacher, I saw in practice how they give students real hands-on experience with biotechnology so that teachers can show students how those biotechnology tools can be used to solve real-world problems. And I'm excited to share one with you today. This lab enables students to assume the role of a public health scientist, tasked with tracing the source of a food poisoning outbreak that occurred at a food truck rally. When I was teaching, I always tried to find activities where students were solving a problem or working through a mystery because it really boosted student engagement. When my students were invested in the activity, it helped them learn the underlying concepts. Today's experiment allows you to introduce a number of biotech methods to your classroom. These include PCR, restriction enzyme digestion, and gel electrophoresis, all through an engaging case study approach. It's a great way to teach all of these essential techniques in an easy to follow, step-by-step manner. The lab offers flexibility depending on your class schedule. It's designed to be able to be run within 3 45-minute class periods or 2 90-minute class periods. And towards the end, I'll explain what I mean with that. Because this lab includes all of these techniques, I'll be showing each one so you can see what's involved. But much like a cooking show, I'll immediately continue on to the next step without waiting for those incubations, because I don't think you have quite a few hours to spend with me. I'm going to start reviewing the background of the scenario and as it's time to share the techniques, I'll switch over to my bench cam and show you how easy it is to implement this lab into your classroom. This lab is great for a wide range of classes, including Gen Bio, STEM, and health science classes, because it allows you to do a number of things. It allows you to, in general, teach the fundamental biotechnology concepts of PCR, restriction enzyme digestion, and gel electrophoresis with robust, reproducible results. It allows you to engage students in a novel public health mystery, and it allows you to review essential aspects of experimental design, including the importance of controls. We all know food poisoning is caused by ingesting food that has become contaminated in some way. The most common cause of foodborne illness is contamination with pathogenic bacteria, including pathogenic forms of E. coli, such as the O157H7 strain. Food contamination with the pathogenic E. coli, O157H7, starts out in the intestines of healthy livestock. E. coli does not harm the livestock, this pathogenic strain. But when cattle defecate, they release this pathogenic E. coli in their feces. Meat can become contaminated with fecal bacteria during the production of ground meat. This is why it's important to cook foods to safe temperatures, because it can limit or eliminate bacterial growth in meat products. Crops can be exposed to feces through improperly processed manure-based fertilizers or tainted irrigation water. This is why it's important to thoroughly wash fresh produce, because it can help to rinse away those pathogens. Now, E. coli are found just about everywhere, and most strains of E. coli are not harmful to humans. In fact, humans have many strains of E. coli as a part of a healthy gut microbiome. However, ingesting the pathogenic E. coli, O157H7, causes gastrointestinal symptoms. This is due to the fact that it produces a toxin that damages the lining of human intestines. Ingesting this pathogenic E. coli does not necessarily mean that a person will get sick. And even if they do, most recover quickly. However, a percentage of people who ingest pathogenic E. coli will experience serious medical complications, which can require hospitalization. In our scenario today, there was an outbreak of foodborne illness at a food truck rally. When tested, the symptomatic patients all tested positive for the presence of the pathogenic E. coli O157H7 in their gastrointestinal system. The students take on the role of a member of a local public health and safety team. Their job is to identify the source of the bacterial outbreak. First, it's critical to determine the source of the outbreak by determining which food truck or trucks the people who got sick ate at. And then, once we've determined the likely source or sources of contamination, DNA testing will be used to confirm the presence of that pathogenic E. coli. The first thing students see is this pre-lab activity. They're told that 10 attendees were surveyed about the food trucks they visited. Importantly, the survey included both those who were sick with foodborne illness and those who were not. The students used the data like a logic puzzle to determine the likely source or sources of contamination. Before I show you the table, I want to share that I love this part of the scenario. Rather than just give the students the information, the lab asked students to practice their critical thinking skills and really examine the data. The students then become more immersed in the scenario when they're discussing in their groups. And boy, I've seen it, they can get feisty. As a teacher, it's a great way to make students think. I'm sure you've already guessed, it's not a simple answer. And it's a lot more like what a public health officer might see in this situation. So here is the data that students are given. They're then asked to determine the possible sources of the E. coli exposure. As you can see at first glance, there is no one food truck that can be determined to be the obvious source of the contamination, as there is not a single one that all of those with foodborne illness visited, that none of those without foodborne illness did not. The students are guided through a series of questions to help them make their determinations. These include, are there any people with foodborne illness that only visited one truck? Is there a single food truck visited by every person with symptoms of foodborne illness? Are there any food trucks that none of the people without symptoms visited? They're then asked to determine the likely sources of the pathogenic E. coli. Now, eventually, they'll determine that the interview data suggested that the people were exposed to the E. coli O157H7 at two possible sources, either the burger barn or the sandwich shop. The health inspection results from both trucks showed that they used all of the proper food storage and safety procedures. This suggests that the source of the contamination was not the food trucks themselves, but rather ingredients that were contaminated prior to their arrival at the food trucks. Upon further investigation, it's determined that the two food trucks share suppliers for two ingredients, the lettuce and the tomatoes. So in order to prevent further foodborne illness, it's critical to determine which of these two ingredients was the source of the pathogenic E. coli. So now we've determined the possible sources of contamination. So we're going to use DNA testing to determine which of the food samples is the source of that pathogenic E. coli. This type of DNA testing typically follows the same set of steps. First, DNA is isolated from the suspected sources of contamination. In this case, the DNA has already been isolated from the lettuce and tomatoes, which could be the source of the pathogenic E. coli. Then, polymerase chain reaction, or PCR, amplifies a section of DNA that could be used to differentiate between non-pathogenic and pathogenic bacteria. For this experiment, PCR will be used to amplify a 400 base pair segment of DNA that differs between the pathogenic O157H7 strain and other non-pathogenic strains of E. coli. This is critical, as E. coli are abundant in our environment, and we would expect there to be some bacteria on any raw produce we test. What is important is that the strain of E. coli that we see in our samples is whether it is the pathogenic O157H7 strain or whether it's a non-pathogenic strain. We're only concerned if we find that pathogenic strain, as that is the one that caused the food poisoning that was experienced at the food truck rally. I remember the first time I used a mini PCR lab with my students. It had been a while since I had done any lab work. But the great thing about the lab guides is that they have everything you need to prep this lab and to run it with students. The instructor guides have the students set up for each phase of the experiment, and student guides are specific about what students need to do when. Our customer support is also always available for you to email or call during business hours if you have a question. Our goal with all of our labs is for any teacher, regardless of their biotech experience, to be able to bring hands-on biotech to their classrooms. And we're here to help you make it happen. Now, one of the ways we've done this is that the kit actually comes with prepared DNA samples, which greatly increases the likelihood that the student's PCR will work. The DNA samples are simulated, and so there's absolutely no pathogenic risk. There is no live E. coli. The lab also comes with control samples, so you can emphasize the importance of good experimental design, and your students have clear reference results they can use to interpret their samples. So we have control samples from the non-pathogenic E. coli and the pathogenic E. coli for students to compare. I'm going to switch to my bench cam to show you what goes into preparing the PCR samples. I'm going to use the mini PCR thermal cycler to perform this step, but I want to make it clear that if you have a thermal cycler at your school already, you can absolutely use that instead. The first thing I have set up is my PCR samples. I still have all the components in their original tubes, but for student setup, your students would receive these samples in labeled tubes that you've already aliquoted. The samples can be aliquoted up to a week before the students perform the experiment, and you can set them out at the beginning of class because the reagents are all stable at room temperature for extended periods. The first thing I'm going to do is go ahead and use that proper safety and put on my gloves. So as I said before, we're going to have 4 samples that the students are going to prepare. I've already labeled the tubes. I have them here in my mini rack. I love the mini rack because it allows the students to see the bottom of the tubes, and so they can see whether everything is about the same volume, which is really useful as they're setting up this PCR. So again, my four samples are the non-pathogenic control DNA, the pathogenic control DNA, the lettuce sample, and the tomato sample. The PCR is going to amplify a small section of DNA that is different between the pathogenic and non-pathogenic E. coli. The great thing about PCR is we only have to add 3 things to our tubes. This makes this lab fantastic for students performing PCR for the first time. The lab guide takes students step by step through what they have to add and makes it super easy for students to be successful. So I'm going to follow the lab guide, and the first thing I'm going to add is the master mix. And so this is, you can see it's got this green color. The master mix contains essentially all of the general ingredients required for PCR. So it contains the polymerase, Taq polymerase, which is the enzyme that copies the DNA. It contains the free DNA nucleotides. It contains buffer. And this particular master mix also contains loading dye. So this is great because once it is time for us to um, take these samples and load them on the gel, they're already ready because they've got that loading dye. Now, just a fun trick I always use with my students, I have them put their tube back one position on the rack that they were using so they know they're done with it. Even if they're using just regular tubes that have an MM on it, I have them put it back. That's just a trick that I had my students use when we were in the classroom. Again, now I want to show you the mini rack, and you can kind of see how they all have that green tinge to it. But for your students, they can pick this up, they can check it out, they can look and make sure that all of their samples have their loading dye or the master mix in them. So I did not change my pipette tip with a master mix because there wasn't anything in the tubes. Now I'm going to change my tip between each of my samples because I don't want to contaminate either my tube, or I don't want to put the wrong thing or bring something from this tube into this tube. So we're going to change tips each time now. So I'm going to change my variable volume micropipette to the correct volume. And then I'm going to add my primers. So primers are small pieces of DNA that bind to the template DNA, and they flank the DNA sequence that we want to copy. Here's another trick, just because we know students can sometimes get distracted. So what I'm going to do is again, I'm going to switch tips, is I'm going to move that lid. I'm going to kind of rotate it so that they're in that upright position, whereas I had them sideways before. That tells me that I have added my primer to each of those samples. I'm going to turn this one. There we go. I'm also, again, because I'm done with the primer, going to move it backwards. So now I'm going to add my template DNA. This is the last thing that I need to add to my sample. The template DNA is the DNA that we're going to be copying from. So this is the thing that is going to be different in each tube. So what I'm going to do after this is I'm actually going to close each tube when I'm done. Again, the 4 samples are the non pathogenic DNA control. And I'm going to add that directly to the liquid. I'm going to close my tube, set it backwards again, and then immediately close this first tube. You can hear them click when they're closed. So it does take a second, but it just is a great way to teach students how to make sure that they've added everything they need to. Again, put that back. So this is my pathogenic controlled DNA. And now my two experimentals, my lettuce DNA. And my tomato DNA. Put it back. Students can pick up the tube if they want to as well. And then again, just close it. And that's it. That is all of the things that I needed to add. So the PCR setup only took me a few minutes, and it was as easy as following a recipe. The students can see exactly what they need to add in the table in the student guide. So now I've closed all the lids. Now what I'm going to do is mix the samples. This is important to ensure that all of the components are equally distributed because we want to make sure that that Taq polymerase can get to the DNA, that the primers can get to the DNA. So all I'm going to do is just flick them a couple of times. And then to make sure everything's down at the bottom, I'm just going to flick my wrist a little bit to make sure. The other thing students can do, again, is just double check all of their samples, make sure they're about the same volume. So once I have mixed, I'm going to go ahead and actually put them in my mini PCR machine. You'll notice I have some samples already ready. So those are ones that I set up earlier. Again, this is my cooking show demonstration. So I'm going to go ahead and do this and put them all in. Now, if your students have more micropipetting experience, we do suggest using this mixing method where you just flick the tube. Students can also pipette up and down a couple of times. So you have both options. Again, you know your students better than we do, and so you know if that's a good idea in terms of their experience micropipetting. So I'm going to go ahead and take these samples out. Oh. And I'm going to go ahead and close my mini PCR. And now I'm going to go ahead and set up the PCR program on the mini PCR app. Before I do that, of course, my mini PCR needs to be turned on. So I've got it plugged in, and I'm going to go ahead and turn it on. And you can see the lights start to blink. I think you can see, yeah, you can see this blue one starting to light up. And so this is going to tell me that my mini PCR is on and ready to connect to my device. So now that my samples are in the mini PCR, I'm going to go ahead and program the mini PCR using the mini PCR app, which you can download on our website. The first thing I want to do is make sure that my mini PCR is connected. So I'm going to go ahead and click on devices, and you can see that I do have the one that I'm going to use connected. Now I'm using my computer and Bluetooth connection in this instance, but you can connect to a wide range of devices. I regularly actually use my phone to program my mini PCR. And if you need to, you can hardwire using a USB connection. So the next thing I'm going to do is I'm going to set up my program. I hit the plus button and I want to run a PCR. So I'm going to click PCR. I'm going to name it food truck PCR. Now what I'm going to do is input all of the temperatures and times detailed in the lab guide. So all of this information can be found in the lab guide so that students can use that information to set it up. You can even ask them at this point to pause and you can double check that they have input all of the times and temperatures correctly. Once you've double checked it, the students can just click save and run because I've already connected my mini PCR. So at this point, once the program has started running, the mini PCR will complete the run even if it's disconnected from a computer. So if your students need to leave because class is over, if you've got that 45-minute class period, they can just leave their mini PCRs running and the program will complete. We even have an auto start feature. With that, you can pre-program your mini PCR if your school doesn't have a one-to-one setup. For me, I actually, for the first couple of years, needed to bring in a computer cart so that the students could program the mini PCR. But there are lots of different ways that you can set it up so that it will run the program. Once the program is complete, the samples are stable at room temperature for extended periods. So as a teacher, you don't need to be paying attention to how long they've run and then worry about quickly taking all the samples out and putting them in the fridge or freezer. Even if the students are coming back the next day, the amplicons are completely stable again at that overnight room temperature. The PCR protocol takes a second to get started, but you'll see that once it does, there's an animation that accompanies each phase of the PCR. So right now we're looking at that initial denaturation. That is going to be that high temperature where we're separating the two strands of our original template DNA and breaking those hydrogen bonds. I'm going to let you watch the program through a single cycle of denaturation, amplification, and extension so that you can see the accompanying animations. But one of the things that I really love about the app is it allows students to really see what's going on at each step. So here, we've already gone through the initial denaturation of that first 30 seconds, and now we're in the annealing phase. So we spent 7 more seconds denaturing, and now we're annealing. This is the phase where those primers are going to come in. They're going to bind to the two ends of the sequence that you're looking at. And the temperature is going to come down to allow those hydrogen bonds to reform. So you see the two primers coming in so the students can really pay attention to what's going on. You'll also notice that there's a graph. That graph gives the expected number of DNA copies that you're going to have of this small region of DNA that you're amplifying. Eventually, as you go through multiple cycles, the students will see that this is an exponential curve. So with every cycle, the students are going to see that doubling. Now you saw the Taq polymerase come in. The Taq polymerase is going to amplify those regions and extend from the primer and make another copy of, again, that region that we're looking at. So now we're on our second cycle, and again, we're going to see everything happen again. And you see that the DNA copies increase to 4. So I'm going to go ahead and let this run while we are looking at what's going on in between, and we'll go to our next step. So in this lab, the PCR is used to copy a 400 base pair segment of E. coli DNA that is present in both strains, but that contains a short sequence unique to the pathogenic E. coli O157H7. Following the PCR, we're going to perform a restriction digestion. This is going to allow us to identify the presence of that pathogenic E. coli. Restriction enzymes are essentially molecular scissors. They recognize and cut a specific short DNA sequence. So restriction enzymes, as I said, act like molecular scissors. They recognize a specific short DNA sequence and cut at that sequence. The pathogenic E. coli have a sequence that a restriction enzyme recognizes in this case. I'm going to show you how to add the restriction enzyme to the samples you saw me take out of the mini PCR earlier. So the next thing students are doing again is that restriction digestion. And the first thing they're going to need to do is transfer each 15 microliters of their PCR sample to a new tube. I'm going to go ahead and kind of move things around. And again, I already have my tubes set up. So these are labeled R1, 2, 3, 4 for restriction digestion. So I'm going to go ahead and again get that volume correct on my variable volume micropipette. Right-handed, it's much easier this way. All right. Now I'm going to go ahead and take 15 microliters of that PCR that we just did and add it to this tube. Again, move that tube lid so that I know that I've already added it if I'm just looking at it from the top. So you will have a little bit of that PCR sample left, and that is okay. The students won't be transferring all of that volume. Again, switching my tip between each sample so that I know that I have only transferred what I need. So I'm going to go ahead and check those. And again, I do have sample in all 4 of my tubes. So I'm just going to move away my PCR samples. At this point in the lab guide, we do instruct the students to bring their tubes to the teacher, and the teacher is going to add 2 microliters of diluted restriction enzyme to the samples. So in the teacher guide, we go over how to dilute the restriction enzyme. And you can do that a few days ahead of time. So now I'm going to take the restriction enzyme is a very small volume. So that's part of the reason why we want the teacher to do it. So we're going to take that 2 microliters of our restriction enzyme. Remember, this restriction enzyme is going to recognize that pathogenic, or is going to cut that pathogenic E. coli. So again, because this is the only other thing I'm adding that sample to the existing liquid, and I'm closing the tubes, which is sometimes a little bit hard with that, but I can do it, to make sure that I know that I have added that sample to each of these tubes. Again, so I've closed all the tubes and made sure that I've added that restriction enzyme. So You can, have the students hand you each one and then they can close it as you're adding it to the next one. Again, just like last time, we want to make sure that that restriction enzyme is um, distributed in all of our samples. All right, so I'm going to pull out my other mini PCR. Now you obviously, you're going to use the mini PCR that you used. I just, again, cooking show style, have mine already set up. So again, I've mixed them, and then I'm going to put them in my mini PCR. I'm going to pull out the ones that I've already done and go ahead and close it and turn it on. So again, you see the lights start to blink, and that tells you that it's ready to be hooked up to your device for the heat block mode this time. So I'm going to go over how to do that, and then we'll let these go overnight. So again, my restriction digestion is already in my second mini PCR. So now we're going to go ahead and set that up. Again, I'm going to make sure that I am connected. I now have my second mini PCR connected. So I'm going to go to my library, and this time I'm going to select the heat block mode. The heat block protocol is for 37 degrees. And again, I'm going to go ahead and name this food truck RE, restriction enzyme digestion. You have a couple of options with your restriction digestion. We recommend that you allow the digestion to incubate overnight for complete cutting. And so that's what I'm going to put in my app. I'm going to go ahead and just say 24 hours. This will allow the mini PCRs to cut overnight. And when the students come in the next day, they can just stop the program and pull their samples out. However, you can incubate for as little as 15 minutes. The lab guide includes information on what the results would look like from that 15-minute digestion. The DNA will most likely not be completely cut, but you absolutely can still interpret the experimental results definitively if you use that 15-minute digestion. So again, just like before, I'm going to click Save and Run. I'll select my other mini PCR. And so now this again is going to maintain that steady temperature of 37 degrees. In this case, we're not seeing those animations or the graph because we're just maintaining a steady temperature. But I'm going to let that go ahead and keep going while I go back to my PowerPoint and tell you what we're going to do next. So once again, the restriction enzymes recognize a unique DNA sequence. In this case, the pathogenic E. coli contain this unique sequence, which allows the restriction enzyme to cut the 400 base pair product into two bands of 250 and 150 base pairs. Now, the non-pathogenic E. coli do not contain this unique sequence. So the restriction enzyme will not cut the DNA, and the 400 base pair sequence will remain intact. Because we have these different size pieces of DNA, and there are differences in the sizes of the fragments from the pathogenic and non-pathogenic E. coli following restriction digestion, we can use gel electrophoresis to determine which food sample contained that pathogenic E. coli that caused our foodborne illness at the food truck rally. As I said, we're going to use gel electrophoresis to visualize the results of our DNA testing. I'm going to load those samples that have already undergone the overnight restriction enzyme digestion on the gel, and then I'll come back and explain what we're looking for. I'm going to load my samples onto the gel now, and then we'll go over how this will allow us to analyze the results of our experiment. Let me go ahead and move my PCR machine. Now I'm going to bring in my blue gel. So I'm going to kind of set up my space to be able to bring in my blue gel. So I used sea green tabs to pour my gel for our blue gel all-in-one gel electrophoresis system. So I'm going to go ahead and bring over, this is my casting tray. So this is what comes with the blue gel, and you'll use this to pour your gels. Today I used a 2% gel, but all of our labs tell you exactly what percentage you want to look for. I poured my gel using our all-in-one agarose tabs. When these came out when I was a teacher, I almost cried tears of joy. For anyone who has spent any time prepping gels for students, you know how long it can take to weigh the agarose powder and then add the TBE and then microwave them. And so it took a lot of time for me to just, do all those steps. The all-in-one agarose tabs, one tab is 2% gel that you pour with the blue gel. So when these came out, it saved me so much time prepping for my labs. I also eventually actually let my students pour their own gels. For me, it was really important. I was teaching a biotech class. I wanted them to be able to take a lab from start to finish. So my students actually were able to use these to pour their own gels. And again, it just made it super easy for them to be able to pop a tab in an Erlenmeyer flask, add some distilled water, let it dissolve, and then put it in the microwave. I had microwaves in my classroom that were just for biotech. So once all the agarose crystals have gone into solution, then you pour the gel. So I, showed the students how to do it a couple of times, and then, then I do, then we do, every group. I watched them pour the gels, and then from then on, anytime we were doing a lab, they were pouring the gels themselves. So I used our 5-well comb when I poured the gel. So this acts like a mold to make my wells for my samples. We actually have a number of different size combs that come with the blue gel depending on your needs. So we have the 5 and the 6. We also have um, the 9-well comb and the 12-well comb. So the 9-well would be great if you're having two groups share a blue gel where they would just put their ladder in the middle and then their samples would go on either side. So all these come with the blue gel. You actually get 2 of each and you can store them on the back of the casting tray. So I'm going to go ahead now and open up my blue gel and put my gel in there. It's great. One of the things that students did before I got mini PCRs is they had a really hard time putting the gels in and making sure that they were in the right location and making sure that they weren't backwards. So one of the great things about these is that there are guide notches that match up with guide notches on the casting tray. And so students, it would be really hard for them to put it in backwards. I'm not going to say that they wouldn't, because we all know sometimes students do things that you don't expect, but it's very hard for the gel tray to actually go in backwards into the buffer chamber. So now I'm just going to add enough buffer to cover the gel. It's about 30 mils, but essentially what you want to do is you just want to make sure you have enough buffer on either side and then just covering that gel. Okay, As I load, I'm going to just share some of my best practices from my years in the classroom and thinking about where my students struggled. The first thing that we're always going to load when we're loading a DNA gel is our ladder. And I'm going to go over what a ladder is in just a couple of minutes, but we always want to make sure that we have a ladder to compare our samples to when we're running a DNA gel. I'm going to actually load this gel using our fixed volume mini pets. These are great. When these came out, I made my students learn how to use these and show me proficiency before I moved them to the variable volume micropipettes because they have the same kinetics. So students still have that first stop, second stop. They use the same tips and they still have that tip ejector. But because they're fixed volume, it teaches them everything except changing that volume. So if you do a couple of just gel electrophoresis labs before you do this lab, it's a great way to teach them how to load gels and also how to use micropipettes. So again, I'm going to load my ladder in the first lane and then my 4 experimental samples in the other 4 lanes. One of the things that I always told my students to do is put both elbows on the table. The goal is because we're hitting such a small target, we want to do everything we can to make sure that the students are successful in loading their sample. So you can't see, but I have both of my elbows on the table. I told my students to try and get their eyes directly above the wells. That again makes them really cognizant of the target, and they want to use 2 hands as well. Students can hold their wrists, students can hold their hands. I actually shake a whole lot, and so I use my second hand to make the micropipette not shake. And so I'm going to do this, and then I'm going to slowly put my tip in, find the well, and slowly um, release my sample. And then the most important part, and something that sometimes students struggle with, is actually bringing your tip out before you release the plunger. I've had students who did great. They got their samples in, but then they pulled it all back out when they released um, their plunger before they had pulled their tip out. So again, we want to make sure that we're stable when we're loading a gel. We also want to make sure that we only just get into that well. So again, I love the 5-well combs because they're a much larger target than the 9-well combs, and so they're a great way to start students. I always go in at an angle. That angle helps to make sure that I'm not just shoving that micropipette tip into my well, and I just wiggle my tip around to make sure that I am loading the sample correctly. The reason that I say we don't want to put that sample too far down is if you, go too far in, you can puncture the bottom of the well, which obviously will make your sample come out. Or I even had students try to inject their sample directly into the gel. And if they're not at the same location to start, if they're not straight across when they start, then you're going to see some differences in how they run. See, one of the things that I taught my students, you almost need to just go in, right? Just a little bit. You also want to go very slowly. This is not the place to um, be, super speed racer. That's because if you were to be too quick when loading your samples, they could potentially spill out into the next lane, and then you don't necessarily know what your results are. I said that our samples or our master mix contained loading dye when I was making those. And what I mean by that is the loading dye gives us a dye to follow our samples. It also has some sugars in it to make the sample more dense than the buffer. And so our samples do settle into our wells really well. So I'm going to go ahead and load that last sample. Now all of my samples are loaded. You can see how, they're slightly darker than they were before. Now I'm going to use our Clearview spray. So I'm going to go ahead and give my lid a spray of our Clearview spray and then wipe it off. The Clearview spray and these cloths do come with the blue gel. Now, the reason I'm doing that is that one of the things that can impede us from being able to read our gels is condensation on the lid. So that Clearview spray stops that condensation from forming on the lid. It's kind of like when you're scuba diving and you spit into your goggles. Now I'm going to put my lid on. Again, one of the other things that can happen with other systems is that students put the lid on backwards. So the negative should be here, the positive should be here. But if they put it on backwards, and I'm going to talk about how the DNA is going to run in a minute, but they essentially, would run their DNA off the gel in the wrong way. So the great thing, again, I love the blue gels when I was using them in class. My students were able to get results. Once they learned how to load, they were able to get results just about every time. So these are really hard to put on backwards. So the electrodes are made in such a way that it's really hard to put it on backwards. Again, I'm not going to say that students will never try that, but they will have a really hard time. So I'm going to go ahead and plug in my blue gel. The new blue gels have a USB-C to USB-C connection, and they come with a power supply for the other end, but you can actually run them on specific types of external power supplies. So that's what we do when we go to conferences is we actually run them on external power supplies, which is really great. So if you're in a space that doesn't have a whole lot of plugs, it's a great way to be able to still do gel electrophoresis in your classroom. So I'm going to go ahead and turn it on. You can see that the green light has turned on. This will stay on for the entire time. If the green light turns off, that means that your current isn't running properly. And so that'll tell you immediately, maybe you didn't put in enough buffer, maybe the lid isn't on correctly. So now what I always wait for is for some bubbles to form on that negative electrode. I always have my students look for that and then kind of raise their hand once we had loaded a gel, just so that I could check and make sure everything was running properly. So we're going to let this run. I'm going to talk a little bit about how you would interpret your results, and then we'll come back and see how my gel is run in that time. Now that my samples are loaded, I'm going to go over a little bit about gel electrophoresis. Gels are commonly made using agarose, which is the sugar from seaweed. At the microscopic level, the agarose forms a web. The web acts like a size filter as molecules move through the gel. DNA is negatively charged, so when an electrical field is applied to the gel, the DNA will travel toward the positive electrode. The way that I described it to my students was I wanted them to think about a toddler running away from their parent in a crowd. I always had them close their eyes and kind of paint the scenario. They all would tell me the exact same thing. The toddler could move much farther in the same amount of time compared with the mom. Then I had them think about pieces of DNA. It's kind of the same thing. Just like the toddler can fit in holes in the crowd that the parent can't, and the toddler gets away faster than their mom, the smaller pieces of DNA move through the agarose gel faster than the larger pieces of DNA. So here I've schematized a smaller piece of DNA and a larger piece. Again, the smaller piece of DNA has an easier time moving through the web and will travel faster, while the longer piece of DNA gets caught up in the web and travels more slowly. When we started, you saw me load my gel, and all of the DNA molecules were put into the well. But as the gel runs, the DNA molecules are going to separate into bands based on the length, with the longest pieces of DNA traveling the shortest distance and the shortest pieces of DNA traveling the farthest. As I said, in that first lane, I loaded a ladder. A DNA ladder is a series of pieces of DNA of known length. By comparing our DNA in our sample lanes to those in the ladder, we can determine the size of the bands. Here, we see the top one is about 700 base pairs, while the bottom one is about 400. Within the lab guide, students are taken through the process of gel interpretation so they can accurately read the results of their gel electrophoresis. This is such an important step. I would review this with my students before they ever ran a single gel to make sure that they were able to correctly interpret the results. These slides come directly from the classroom PowerPoints available on our website and are particularly useful for reviewing the material. We're going to practice with a few of these types of questions that students will encounter in the lab guide. We want to make sure that students are able to correctly identify what bands they're looking for. So if the samples do not contain pathogenic E. coli, then the restriction enzyme will not cut the PCR product. And so what would that look like on a gel? Well, we'd see that single band at 400 base pairs. Samples that contain pathogenic E. coli will contain a PCR product that can be cut by the restriction enzyme. So what will that look like on the gel? Well, we'll expect to see two bands, one at about 250 base pairs and one at about 150 base pairs. Once you're comfortable with the ability of your students to properly interpret their gel electrophoresis results, they'll be able to compare the results from the lettuce and tomato to determine where the pathogenic E. coli were present. Here's a time lapse of the gel results after about 20 minutes on our blue gel system. I love how the blue gel can be used to visualize results so quickly. I used to have students take time lapses of their gel runs in class, and they had to turn them in with our post-lab questions. Then I knew at least one of them didn't have their cell phone on them. I'm going to show you the results. They haven't run quite as long as the samples ran in this time lapse, but it will allow you to see what the gel looks like in this amount of time. All the bands in the DNA ladder might not have separated, but that's okay. You only need the bands in the relevant range of the ladder to separate enough to interpret results. Remember, we're looking for bands at either 400 base pairs or 250 and 150 base pairs. So now I'm going to switch over and show you exactly what my gel electrophoresis results look like after this amount of time. So I let the gel run as we were going over the background. And I just wanted to share what I see after this amount of time. So I'm going to go ahead and take off my gloves and show you what you can expect to see after, again, just this short amount of time that I've been talking since I loaded the gel. So I'm going to go ahead and turn on the trans illuminator. The blue gel has the integrated trans illuminator, so students can check how the gel is running, and also it's super easy for them to take pictures of their results. They don't have to move the gel anywhere, they can just view it right here. So you can see I have bands in all four of my experimental lanes, and my ladder again has separated about this much right here. We'll go over the results in just a minute, but I wanted to make sure that I gave you an image, or kind of showed you how the gel ran in just that amount of time since I loaded it. We're going to look at a picture of the gel to review the results interpretation now. So I'm going to go ahead and turn off the transilluminator and show you a picture of the gel that has run for just a little bit longer than shown here. As expected, the non-pathogenic E. coli control exhibits a band at 400 base pairs, while the pathogenic E. coli control has two bands, one at 250, one at 150 base pairs. These give our students examples to compare their experimentals with. So now let's look at those. The lettuce sample only shows a band at 400 base pairs. This corresponds with the non-pathogenic E. coli. This means that the lettuce was not the source of the food poisoning outbreak. But in the tomato sample, there are two bands, one at 250 and one at 150 base pairs. This means that the pathogenic E. coli was detected in the tomato sample. Following this result, we know that the tomato was the source of the contamination. Students are asked what next steps they would take as a public health official. So students have gone all the way from determining the source of the outbreak in terms of what food truck or trucks might be the source of the contamination, all the way to the conclusion of the DNA testing and thinking about what would happen beyond. This lab allows you to show that molecular techniques can be used to detect specific pathogens, and students were able to identify the likely source of that pathogenic E. coli O157H7. Based on a real-world scenario, the Food Safety Lab allows students to solve a public health mystery. And I personally think it's an excellent way to introduce those fundamental biotechnology techniques, including PCR, restriction digestion, and gel electrophoresis. On this screen, I've snapshotted a page of the lab guide. I want to show you how our lab guides are written because I think that it makes it so much easier for students to understand. Here you see that the information is broken down into easy to read units, and lots of information is presented visually. You can see here how the concept of restriction and digestion and those cuts are presented. The lab also has assessment questions interspersed throughout to make it accessible even to students who aren't ready for dense text-heavy background. The lab kit is only $88, which is impressive given all of the reagents you get and all of the techniques your students get to perform. I want to reiterate, you can use any thermal cycler and any gel electrophoresis system with fluorescence visualization. I use the mini PCR and the blue gel, but you can use what you already have in your classroom. You'll need about 30 minutes of teacher prep to dispense the samples and make the gels, especially if you're using the all-in-one agarose tabs. Those are a lifesaver. All of the information for performing all of these techniques for what the instructor is doing is in the instructor guide. The lab is set up to take about 3 45-minute blocks. One to introduce the lab and set up the PCR, one to set up the restriction digestion, and one to run the gel. It can also be performed in 2 90-minute blocks because the PCR only takes about 45 minutes. So you can set it up and the restriction digest up in one class period. One of the best things about mini PCR as a teacher is that all of the curriculum is available to download right now. All you have to do is go to the Learning Lab website and click on Curriculum Downloads. There you'll find the instructor's guide and the student's guide. As I said, the instructor guide has all of the information for you to prep the lab, everything from tube labels to how to set up the student workstations at each step. You'll also find the classroom slides. The slides for my webinar today were derived from those classroom slides. They're editable PowerPoints, so you can make them align with your classroom needs. Our latest additions are the e-worksheets and the standards alignment. The e-worksheets are editable Google Docs that you can either just take and upload directly to your LMS or change to fit your classroom. The standards alignment gives you a detailed alignment to NGSS, AP Bio, and TEKS. These are great for determining how this lab fits in your curriculum. I want to thank you so much for taking part in this webinar today. Please feel free to reach out to Mini PCR if you have any questions. Have a great day. Bye now.